Greetings, students. Mr. Sanger, live from my living room, and I want to do a quick screencast to introduce you to this labish activity, which is kind of a paper pencil look where we're going to do fruit fly genetic crosses. So here's a lovely picture of a fruit fly and an inspirational quote about fruit flies to get you in the mood. So the objectives of this activity, you're going to analyze data from various crosses to determine the mode of inheritance. Is this just a simple dominant recessive autosomal trait? Is it a sex-linked trait? Is it a monohybrid? Is it a dihybrid? Is there linkage involved? What's going on? You're going to try and infer that from data from crosses. We're also going to review and practice the use of the chi-square test. So note that this is paper pencil, but I do want you to kind of appreciate where the data came from and understand how these crosses are actually done. If you do go on to study genetics in college, it is quite likely you will be carrying out these procedures with actual fruit flies. It's a very common intro biology lab in college, and it's kind of fun to do. So how's it going to work? So background. First of all, this is a shocker to a lot of students. Fruit flies with specific phenotypes can actually be ordered from a catalog. You can just call up Carolina Biological Supply and be like, hey guys, can you send me some ebony body vestigial wing flies? And they're like, yeah, sure, no problem. And they come in a vial that looks like this. This little goopy stuff at the bottom is actually the food. This is just kind of like a foam plug that kind of keeps them all in there. And then along the wall, you can see there's these little like larvae. They metamorphose, as we'll discuss in a moment. And so these are just little larva cases in there. And then there's little fruit flies buzzing around. And so that's kind of how they show up. Um, so within these vials, the fruit flies are going to continually mate and have offspring. So here's the male. Males are a little bit smaller. They have this kind of black bottom. That's what helps you identify them from the female. And so they're going to continue to mate. They're going to make an embryo. And then that embryo, it looks like a little worm. And so you can see these little worms crawling around in that food supply. They grow, and then eventually they climb up onto the wall and form this little case. We call it a pupa. And it's much like a butterfly. A caterpillar turns into a butterfly kind of thing, only in this case, it's this little like worm-like maggot thing that turns into a fly. So that's their life cycle. And so they're going to continuously mate. This is a little PG-13 image. Actually, that'd be rated R. X even. Okay, so anyway, that's kind of what is happening. They're mating and they're producing these offspring continuously. Now, in order to work with these flies, we need to be able to kind of select certain flies to be mated. And so what we do is we use a tool, I kid you not, it is called FlyNap. And it's this little liquid that basically knocks out the fruit flies. It puts them to sleep. And so you get these little brushes, you dip the brush into the liquid, get the liquid on there, and then you just kind of squeeze the that brush in between that little foam cork I pointed out, and you just kind of let the end of that, it's basically like a fancy Q-tip, sit inside there. And then the gas builds up and the flies kind of, and they fall asleep. They are knocked out. What you can then do is you can dump the fruit flies out onto a note card, and you use a paintbrush, just like your little color by number paintbrushes you used when you were a kid. And you sort of look at the fruit flies through this microscope, and they look like this. And then you can kind of sort them out. So you look at the abdomen, there's that black bottom there, so that's a male. This is not black bottom, so that's a female. And you can sort them out based on male and female and based on phenotype as well. Okay, black body go this way, vestigial wings go that way. And you just kind of sort them however you want. And what's kind of fun about this is that FlyNap, it's temporary. So the flies eventually, as you're doing this, if you're too slow, the flies start to wiggle. And you're like, oh no, they're waking up. And then every now and then one will like, and they fly away and like, and you lost one. So it's a very intense experience trying to sort these fruit flies before they wake up. So after you've sorted them, you take flies with the desired phenotypes and you mix them in a brand new vial. So if you want like a red-eyed male and a white-eyed female, great. Take your red-eyed male, your white-eyed female, and put them together in a new vial, and then you're going to begin this process of mating and producing offspring, and then you're gonna get offspring, and then again, you knock those offspring out, and you can count how many of each type are in there to get your ratios. So that's basically the background for how the data you're about to look at was acquired. So you're going to analyze data from 4 crosses, and those crosses involve a number of phenotypes. You're going to see gray body, which is kind of the typical normal fruit fly color. But then there's this variation, the black body. They call it an ebony. It's kind of a pretty black fruit fly. Vestigial wings mean their wings don't work anymore. They're these little kind of spindly little wings versus the normal wings. Eyeless, that's a little creepy. They literally have no eyes, aptly named. And then red eyes, which is the typical color, and then white eyes, which is another one. So you're going to have these little phenotypes, and these are all mutations that geneticists have stumbled upon, or they actually used to like irradiate fruit flies to give them mutations, which is kind of mean. So these are all fruit flies that you can just pull up at a catalog and order and play around with. You can expect the following types of outcomes in your data. So of the 4 sets, you're going to see complete dominance. In some cases, there's going to be a monohybrid one and a dihybrid one. You're going to see a sex linkage case, and you're going to see an autosomal linkage. And the key is you're going to look for clues in the ratio of phenotypes of the offspring or the types of offspring to figure out the mode of inheritance. Okay. So for example, maybe you count your offspring and you see, oh, there's like 74 of one type and 28 of another type. Well, that's pretty close to a 3-to-1 ratio. If that's the case, well, 3-to-1 ratio, that tells us that it must have been 2 heterozygotes that were crossed to give us that 3-to-1 ratio. So you kind of use the ratio to sort of go backwards and figure out what the parents were. Now, of course, if it's, 74 and 28, that's pretty close to a 3-to-1 ratio. But how do we know if we're close enough? Well, that's where our chi-square test comes in. And we've already talked about the chi-square test and how to use it. I believe it was in the when we talked about mitosis. So if you need to review chi-square, you can go back and watch that screencast on how to do that. But it should be on the formulas and equation sheet for AP Bio. And you're going to use that chi-square to determine if the numbers you get are close enough to a 3-to-1 ratio to be considered statistically significant. And that's basically the activity. So again, we're engaging in these science practices of AP Biology. So at this point, you're going to look at the data, do your chi-square, and that's it. So good luck to you, children.